Please welcome Dr. Zhang.
Hi, everyone. I'm Dave Zhang. I'm very glad to be here, and I'd first like to take the time to thank Martine and Genesis, and in particular Temple, for putting up with my procrastination. I gave her my slides only about 10 minutes before this started. So, let's see. So I'd like to start by saying that I'm a DNA engineer, and as a DNA engineer, I would like to see this kind of a logo running around amok basically 5 to 10 years from now. So DNA engineering is very different from genetic engineering, and most people here probably know what genetic engineers do. Basically, they use the central dogma, which is that DNAs get converted into RNA, and then that gets converted into proteins, and then you can regulate and control each of the steps along the way. And what we do as DNA engineers is something very different. What we do is we just use the biophysical properties of DNA as a material. And DNA as a material is very nice because it's very well understood. So in our case, we basically build structures and functions and logic components and amplification components, and it's all very nice. And some of you might ask, why do you want to do this? And my simple, short answer is because it's easy. It's much, much easier to work with than proteins. Protein folding is, well, by a lot of people know that it's very difficult. And under most certain assumptions, protein folding is actually an NP-hard problem. And for computer science in the audience, that knows that it's a problem that will never get easy. So assuming that that's the case, then if we can build everything with DNA, with nucleic acids, not necessarily just DNA, but also RNAs and PNAs and LNAs and all of these nucleic acids, and if we can use these materials to build the exact same functions that proteins do, then it might be a very worthwhile alternative to look into. All right, so first I'm going to talk about how we abstract DNA. So DNA, as you mostly know, is this double-stranded molecule, and it has all of these bases involved. Usually in literature, it's basically just show the sequences in GCAT, and then you show 5' to 3' with a little hook at the 3' to denote the 3' end. What we're going to do is we're going to go a step simpler. We're going to abstract these as domains shown by numbers, and with the understanding that these domains can be arbitrary sequences. You can insert whatever sequence you want. They can be biologically relevant sequences, or they can be sequences that you design from scratch using DNA software. And the idea is that everything we develop, everything I'm going to present today, should work just about equally well no matter what sequence you use. And that's very different from ribozyme work and deoxyribozyme work by the artificial selection people. So that's something to keep in mind. So we're not going to show the sequences at all. The dots just means that they're base paired, and then basically you have the domains 1 and 2 here. Now, I said earlier that we can try to do everything that basically nature does and cells do with proteins with DNA. So what exactly do they do? Well, first they build structures, right? I mean, structures are important because it prevents you from becoming amorphous blobs. So basically you have cell walls, for example, and you have cilia, and you also have actin and myosin and all these very complicated structures. Now, the largest of these are on a cell level. Obviously, the largest of these are people like us and blue whales, and they're huge on the order of meters. But in terms of an individual cell, it goes up to about 10 microns, right? And so that's our gold standard, our benchmark. Can we basically use DNA, short, simple molecules of DNA, nanometers in size, to form these things that are microns large? And so this kind of work, basically, we start by building DNA tiles, and these are 4 nanometers by 7 nanometers. And again, I'm going to be using the notation that I used earlier, where we don't show the exact sequences, with the understanding that every sequence works pretty much with this. And basically, we have 2 tiles here. Now, each of these tiles has 4 unique binding sites. They're single-stranded overhang domains of DNA that can bind specifically to their complements, where the bar above a number denotes a complement. So in this case, the 1 can bind to the 1 bar here. And the center part actually just gives this material some sort of structural rigidity, so we can ignore that. So you can actually further simplify this as 2 little tiles that look like this. And what happens when you have lots and lots of these in solution for this system, for this example system, is that they'll come together something like this. Basically, the 3 will bind to the 3 bar, the 4 will bind to the 4 bar, the 1 will bind to the 1 bar, and you can imagine this large periodic structure emerging. And experimentally, when we actually do this kind of a thing, we basically mix it together, maybe 100 nanomolar solutions of these, then we actually formed these huge periodic tiles about 2 microns by 8 microns long. This is an AFM image, and it shows a lattice that basically is comprised of 500,000 tiles. That's 500,000 tiles. Now, I know with the Respirocyte work that we talked about earlier that they're adding on 10 million molecules per day or per hour, I don't remember. But this all happens over the course of about half an hour, and we build many, many, many copies of these. Basically, this is the parallelism of chemistry. It all forms basically all at the same time. And we're not limited to two-dimensional structures either. So this recent work by a group at Purdue University shows the self-assembly of three-dimensional structures. So up here we have tetrahedra, and these are actually much smaller. People are working on making larger and larger three-dimensional self-assembly structures of DNA, but we start them all. So we have tetrahedra, and these are AFM images, and over here on the right are cryo-EM images. And on the bottom here, these are dodecahedra, and yeah. Basically, this, again, uses a very similar motif to the one that I showed earlier. Basically, you use only the complementarity properties of DNA to design and demonstrate these. And in case there are mathematically oriented people in the audience who are wondering if there's some special symmetry property that basically these share, then no, we can also build aperiodic structures. So what else do cells do? Cells basically process information and are being generally good at being alive. So for example, if you're this happy amoeba cell and you happen to, could we get rid of that, please? You happen to see this nice algae steak, then you might be thinking to yourself, I really want to solve the Navier-Stokes equation and move towards it. Well, not really, but you probably do realize that it's some sort of a food as opposed to a piece of granite, and if you see food versus granite, you move towards the food. So basically what I showed earlier with that little cartoon is two separate aspects. There's the logical processing aspect, saying, is this thing a piece of food? What should I do? Should I move towards it? Should I run away? What should I do, right? And the separate task of actually actuating and moving towards is carrying out what you've decided. So first, let me talk a little bit about logical processing. Now, if you're the amoeba, at the risk of becoming too anthropomorphic here, you might first ask yourself when you see something on the horizon, is it moving on its own? If it's not, then you can probably maneuver around it and safely ignore it. If it is, then you have to ask a second question. Is it bigger than me? If it is, then you probably run away, and if it's not, you can start getting out your bib and start eating, right? So in terms of logic, this can be expressed in a form known as Boolean logic. And so the decision, so the decision, for example, to run away is the logical and of moving towards something, or something that's moving autonomously and something that's bigger than you. And the decision to attack is something that's moving and something that's not bigger than you. So this is, again, known as Boolean logic. It's very well studied in electrical engineering, as you all know. And we want to implement it with DNA. So how do we do this? So first, we take 2 strands of DNA. These are arbitrary sequences. These just happen to be the sequences I did experimental work on. But again, these can be mRNA sequences. These can be microRNA sequences. These can be whatever nucleic acid sequences that exhibit Watson's-Crick specific base pair binding. So we abstract these as the 2 domains, and we consider what happens. So we have this substrate molecule, which is this double-stranded molecule with overhangs on one of the strands. And if you only have one of your 2 inputs present, then what happens is that the one domain will bind on, and the 2 domain will migrate back and forth because they're isoenergetic on a phase space. And eventually, this input is released, and it doesn't really do anything. You don't produce any output product. Now, if both input molecules are present, then you get a cooperative binding behavior in which both of these gets attached to your substrate, and you release a second molecule, which we call the output. This is a different sequence molecule from the inputs, and it can be used to do other things. Now, for what we demonstrate, we basically just have a fluorescent reporter so that when this gets produced, then you have an increase in fluorescence. So how well does this work? So if you don't have any of the 2 inputs at all, then you basically get a flat line, which is good. And now if you have a lot of one input and none of the other input, then you still pretty much get a flat line. There's a little bit of leakage. And the other thing I should mention is that all of these are using unpurified strands of DNA. The purity is very, very low. It's only about 80%. So the system if it works, is very robust to stoichiometric and synthesis errors. And next, we tried basically the minimum of the two concentrations being 20 nanomolar. So basically, this is 20 nanomolar and 60 nanomolar, 20 nanomolar and 40 nanomolar, 20 nanomolar and 20 nanomolar, and so forth. And using different minimum concentrations of these two, you get different behaviors. And if you plot these all on a 3D plot, then you see that the amount of output production that you get is roughly the minimum of the two input concentrations, and this basically forms the basis of a logical AND gate. Now, using very similar principles, we can also build a logical NOT gate and combine the two to form a logical NAND network. So again, I'm not going to step through the data too much, but if you don't have any, and we also build a threshold on top of that. So when you combine all of these together, then you get this very nice NAND behavior where you have to have very high concentrations of both to shut off a switch. And for those of you who are electrical engineers here, then you know that the NAND logic gate is a universal logic gate, meaning that you can express all sorts of logic with this basic motif. You just have to make lots and lots of them. So the next part is actuators and motors, basically after you decide what to do. So for example, your inputs last time, which are DNA or RNA, might be produced in an output molecule that can be transcribed in, or some other way of converted into some kind of decision, right? And then you want to actuate them somehow. So again, this is a very simple motif developed by Bernie Yerke, and basically these are called DNA tweezers. And what happens is that you have this flexible junction point here, which can move back and forth. And what happens is that when you bring in a second strand of DNA, you can cause them to basically be, well, pinched up in a closed state. And so you can imagine something like this being like a channel for like a gating element for a channel, for example. And you can actually reverse this process by adding in a complementary strand of DNA and then letting it open again. So this is kind of like a lot of ion membrane channels. And in terms of experiments, then Bernie Yerke is a scientist at Lucent. He showed that you can turn this on and off multiple times, and there's some amount of imperfection in the system, but it works pretty well. And people have built on this work a lot recently. So there was a 2008 Nature paper by the Niles Pierce group that showed an autonomous DNA walker that would walk along a track. Now, what else do cells do? Some of you are probably begging to yell at me that cells reproduce. Cell isn't just like a crystal, right? It makes more of itself. And so, for example, if you have one cell, you get into 2 cells, and you turn into 4 cells. And this happens on a cellular level as well as an organism level. If you have 1 bunny, it turns into 2 bunnies, which turns into 4 bunnies, right? So what can we do? Can we do something similar with DNA? So again, we basically have an arbitrary strand of DNA. How do we exponentially amplify this concentration? Again, we parse this into 4 arbitrary domains. This is a completely arbitrary parsing. And what happens is that, again, we use a different substrate. This thing binds on and releases a molecule it's a waste molecule. And then another strand comes in and binds to this substrate intermediate, and you release an identical molecule to yourself as well as yourself. So this is an autocatalytic reaction. A molecule catalyzes its own production from another similar molecule. So I've summarized the equation here and the chemical reactions here. And if you don't have any of your seed, if you don't have any of your organism or DNA molecule at the beginning, then this reaction doesn't proceed. It actually proceeds very, very slowly at a rate constant of, that's, I think, 5 or 6 orders of magnitude lower. So what happens, if you don't have any of this reaction substrate, if you don't give it any food, then it basically flatlines. Organisms need food to reproduce, right? Now, if you give it a little bit of food and you seed it with a little bit of population, then it grows exponentially until you run out of food, and then it flatlines. And if you seed it with higher and higher concentrations of DNA, then it grows faster and faster from an initial. And so that's all good, and it seems to resemble exponential growth. And in fact, we have proven that it is exponential growth. But the problem is that if you have a huge mix of food, but you don't have any of your initial amplifying autocatalytic DNA, then you still get exponential growth. And so this is kind of like a spontaneous generation problem, right? So that's not very good. So it's not really alive. It's more like crystallization in a supersaturated solution. But the pro of this is that basically it functions as a PCR analog, qPCR analog, right? If you know exactly what you're looking for, you can amplify its concentration dynamically as an exponential process. And in the laboratory, we've been able to demonstrate specific detection of DNA down to 1 picomolar concentration. Now, for those of you who don't know, 1 picomolar concentration is about 1 molecule in 1 eukaryotic cell volume. So basically, if we take that reaction and break it down into something simpler, instead of catalyzing yourself, you catalyze something else. And now we're thinking in terms of biotherapeutic and disease detection purposes, then you can get a linear amplifier too. And in this linear amplifier, you actually modeled it very well. Basically, you can break down this reaction into 3 separate steps. And if we can measure the rates of every single step and then build a model around this. And the dotted lines here are the models, and the solid lines are the experimental traces. So basically, this level of fit is almost unseen in biology. I'm very proud of it. So what happens if you want a good disease detector, it has to be very sensitive as well. Basically, if you have a single mutation anywhere along the line, it shouldn't be amplified. And we basically tried to do this work here. We basically took 4 different positions along the strand of DNA, which was 22 base pairs long, and we mutated it in various ways. We either arbitrarily changed one base to another base, or we inserted a base, or we deleted a base. And basically, this line that shoots straight up here is the normal target, the target that you're trying to see. And all of these lines are basically with 1 base pair mutations. And so you can see the activity gets shunted by a lot. And this data is summarized here. So down here, basically, this is a suppression factor. And it's plotted in log scale. So basically, this is how much slower you are than the correct target. And basically, here's our control, basically, if it's 1. And as you can see, basically, a single base pair mutation of any type, whether it's an insertion or a deletion or a mismatch, usually suppresses it by a factor of 20 or more. And so you can imagine that if you have 2 or more mismatches, because you're looking at different molecules, then you're not going to have any crosstalk at all. So the other thing is that cells interact with non-cells. They interact with the physical environment and pieces of granite. And everything I've shown you so far deals only with DNA interacting with DNA. And so one goal that we've been trying to move towards is interaction with general chemistry, a synthetic chemistry that doesn't have to do with DNA. And so this is work by a group at Harvard University called David Liu. And so basically, he functionalizes a strand of DNA with a little organic group at the end. So in his case, basically, it's this organic group. And I'm not really too much of an organic chemist, so I don't know what this group is, but it's a group. And the DNA is attached by this little bond here. And then you have a different strand of DNA. With a different organic group attached, and they have partially complementary sequences. So when you mix these 2 in solution, then the DNA base pairs come together and form. And now these 2 molecules, small organic molecules, which were in very low concentration before because the DNAs were in very low concentration, let's say like nanomolar range. Now they're suddenly brought very close together, only about 2.4 nanometers across the helix of the DNA, and that represents a local concentration on the order of 1 molar. So basically, you've suddenly shot up your concentration by about 10 to the 9, and that allows the reaction to just spontaneously proceed without any kind of an enzyme. And so basically, you form this product, organic product molecule. And you can actually iterate this process. So I've shown this 2-bar domain here with the intention that you can bring in another DNA molecule that displaces a strand off, brings in another organic group, and you can synthesize larger and larger molecules. I think David Liu's group has actually shown synthesis of macrocycles with like 5 or 6 different steps. So that's about it for my for my talk. So I'd like to remind you about what DNA engineering can be used for in terms of biomedical tools. So you can use it for instrument-free detection using my amplification schemes, and you can also process the logic. If you have this marker and this marker, but not this other marker, then what do you do, right? Like you have an exponential decision tree in terms of the concentrations of relevant biomolecules that you're trying to monitor. And you can try to do embedded control of apoptosis somehow. And I'm not too much of a biologist, but there's a group at Amsterdam, Kurt Goethelf's group, who has been working with using David Liu's chemical synthesis, DNA templated synthesis method to produce oxygen radicals, which in turn causes apoptosis, like Mark mentioned just 5 minutes ago. And finally, an eventual goal is we want to try to control developmental biology. Basically, we have a completely bioorthogonal pathway that we've presented here that can produce nucleic acid molecules, be it DNA or RNA, that can be then fed into the normal cellular machinery, which can change the developmental pathway of cells, for example. And that's, with that in mind, I just have one parting thought, which is something Ray Kurzweil alluded to yesterday, and this is known as Carlson's law. It's the equivalent of Moore's law for DNA synthesis costs, and it's also exponentially getting cheaper. So I really think that this is a method that has potential because of the malleability of this molecule that we're working with, the DNA molecule. And thanks for listening.
Well, thank you. That's really a mind-boggling talk you gave us. Is there a possibility of protein expression from these? That is, can you are there equivalent protein structures from these nanoparticles?
So I'm not really sure what you mean. You can take the output products of the nucleic acid molecules and translate them into proteins. The equivalent of doing exactly what we did in terms of logic and using proteins is very difficult, like I mentioned, because protein folding is very difficult. Yeah, nucleic acid folding basically follows very simple rules, right? And protein folding doesn't. Does that answer your question at all, or?
Well, I mean, it's based on my lack of understanding of the total process, but would there be a protein equivalent of the structures that could be produced?
It's possible, but we haven't looked into it.
Next question, please.
Hey, Dave. Rob Burgess here from Med Nanotech. Great talk.
Thanks.
You know, there's been a lot of focus on atomically precise manufacturing, and I think that what your group, as well as Rothman and Mal, is demonstrating is really a version of molecularly precise manufacturing using naturally occurring molecules like DNA.
Right.
And I was very pleased to see you mention the report on the 3D structures from Mal's group using DNA, and I think the obvious next step here would be to create a molecularly precise motor. And I know you had mentioned the actuators, but I was just wondering if you could comment on the field in general as far as the achieving of a molecularly precise motor by the insertion of a rotor or in a self-powered assembly, and maybe your group's been working on that?
Right. So as far as I'm aware of, there's at least two major groups, or three major groups. There's Chende Mao's group at Purdue that you mentioned, and then there's a group at a professor called William Shi at Harvard, who basically uses three-dimensional DNA origami to build a motor that's asymmetrical, so you can drive it with an AC current. Basically, I guess we don't have a chalkboard here, but basically you have an AC current, so it has basically the motor has two different states, and it's like an expanded state when you try to move it in one direction, and it closes up when you try to move it in another direction, so you can get directional motion driving it with an AC circuit, or AC current. And the other group is Andrew Turberfield's group, which published a paper in Physical Review Letters recently showing a even better molecular walker that follows along a track independently. And the really nice thing about his work is that it uses a diffusible fuel source.
Great. Thanks very much.
No problem.
Next question, please.
Yes, is there any group or your group or someone that it has already managed to or is trying to put the synthetic DNA machines or reporters or and inside cells and trying to make cells do something?
Right. So there are several groups actually who are working on this kind of a technology, trying to use this technology inside of a cell. There's Garrick Seelig, who recently accepted. He was a part of our group, and he recently got a professorship at University of Washington, who's interested in doing putting these inside of a cell. Another work that I feel that I should mention is that I know you work with carbon nanotubes, and actually a couple students of ours, as well in a collaboration with William Goddard at Caltech, have been using making FET transistors using carbon nanotubes with DNA glues between them. So that's also a very exciting work that should be appearing in Science within a couple months.
Any further questions? Not like to thank our speaker.
I have one question.
Oh,
I noticed that you can do microRNAs too,
Mm-hmm.
which you didn't talk about.
Right.
Have
So
you-
we've tested some of these structures with microRNA sequences, and they work.
But you haven't done it in any type of cellular system.
We've done these experiments in cell extracts, and they work. But we don't really have anyone who has a lot of experience with in vivo actual working with cells, so we haven't done that yet.
And does the size of the 1, 2, 3, 4s matter in terms of picking up the microRNAs specifically?
Kind of. There are certain parts of it that matter. So, like for example, the 3 domain in my exponential amplification has to be between 6 and 10 nucleotides. But because you can design everything else around them, then you can arbitrarily parse it so that you have basically you take these 6 base pairs in the middle and call that the 3 domain.
Talk later.
Thank you.